Hi, everybody. Welcome back. This is going to be the mini lesson on cellular immunity. So before we get going into the actual slideshow showing the pictures from the text of this process, I wanted to introduce to you some terms so you kind of get a visual about what they're meaning before we actually jump into their application. So the terms I wanted to focus on are these things called MHC. This stands for Major Histocompatibility Complex, which is a group of proteins that are found on the surface of cells. And there's two different types. There's MHC class 1, and then there's MHC class 2s. All right. So these guys are found on the surfaces of cells, and some all cells, some only a few cells. And they have a different role to play depending on what's going on with the cell. So let's say if you are talking about an MHC class 1, these are proteins that are found on all nucleated cells. So if you have a nucleus, you have an MHC class 1 on the surface of your cells. So I'm just going to draw one here. I'll make it like a little U. So imagine this as being a little receptor. So these are my little key, right? So there's MHC class 1s. They are found on your cell. So if you, say, get infected by a bacterium or you get infected by a virus, you will then put some of that viral or bacterial bit of that antigen in that MHC class 1. So let's say you have a bacteria inside of you, right? So here's the bacteria, and it had all the little antigens on its surface, and you then put some of those antigens in that MHC class 1. What this tells the body, this immune system, as we're trying to figure out what's going on, it's sending the signal with this particular bit of antigen, and the message is, kill me, right? I'm infected, there's a problem, there's nothing you can do for me, here's my signal, kill me, all right? That's the role of MHC class 1s. For MHC class 2s, they're nucleated cells, so just to follow suit here, they're gonna have a MHC class 1. Because these types of cells can be infected as well, but they also are going to have these, I'll kind of put it as a purple box. They're going to have these MHC class 2s. Okay, so these receptors on the surface of their cells. So the types of cells that have MHC class 2s, these are going to be lymphocytes. They're going to be another group called antigen presenting cells. So these are like phagocytic cells, neutrophils, monocytes, your macrophages, your microphages, some dendritic cells, Kupffer cells. Your book has a list of those. So these are immune cells. Their job is to engulf a bacteria or engulf a viral infected cell or pick up cell debris that might have some of the antigenic properties in it. So we don't want to destroy those guys because they're doing their job. So they will put the antigen in this MHC class 1. If they're just, say, just phagocytized bacteria, we don't want to destroy them. This message, so if you have the antigen in this MHC class 2, it will say, help me find some more. Help. I'm having a hard time writing today. So they will, I'm trying to erase. It's not letting me erase. There we go. Help. They're saying, help, look what I found. Don't destroy me. I'm not infected. I ate this. I engulfed it. I'm just showing you its identity. Now you go and you find other things and get activated by me showing you this thing. Okay. So these two types of proteins are what we use, what we describe as antigen presentation. So if you are an infected cell, you will present this antigen in MHC class 1 saying, kill me, I've been infected. If you are a phagocytic cell or some other antigen presenting cell, you are going to present your antigen in MHC class 2 saying, help, look what I found, let's go find some more, destroy it. Don't destroy me, I'm one of the good guys. Okay? All right, so now we're going to get into the pictures from your textbook on this process of antigen presentation. So I'm going to start first with the infected cell. So here you have step 1. Well, let me back up. Let's say you have a nucleated cell. You are always presenting these MHC class 1s, always. That's just part of a natural part of your cellular process. So you're gonna be putting up a class 1 here, class 1 here, class 1 here. If you're normal and healthy, you can have normal self-antigens plopped in that MHC class 1. As the immune cells are coming around and monitoring things, if they recognize a self-antigen, they're going to ignore you. You're fine. They're not going to destroy you because you have normal antigens presented in your MHC class 1. However, here we have a bacteria or a virus. They're showing both, viral or bacterial. It's going to get into the cell. It may be engulfed by a lysosome, may try to break it apart. The virus might be replicating, and either way, in the process of your normal day-to-day activities of putting your MHC proteins up on the surface, you will incorporate some of that viral or bacterial antigen into the surface of that MHC class 1. So you can kind of go through those steps there. You get infected, your Golgi and your endoplasmic reticulum process those viral or bacterial antigens. In your daily, here's my MHC class 1, if there's a bad antigen presenting on that, that's going to tell your immune system that you're infected and you need to be destroyed. That's that come and kill me message for the MHC class 1. Okay. If you are an antigen presenting cell, right, the good guys, you are going to engulf the bacteria. You're going to destroy the bacteria. You're doing your job. You're a good guy. So in your processing, you will only present the MHC class 2s when you are actively processing the bacteria or the virus, whatever you engulfed. So you will be putting up the MHC class 2s only if you have something foreign to express in that MHC class 2. So here we can see the MHC class 2 is a slightly different shape. It's got the antigen bit in it, and it will be telling the immune system, hey, look what I found. Here's its identity. Get activated. Go find some more, right? So that's the major difference between the two. MHC class 1, you're an infected cell, and in the daily process of putting up your MHC class 1s, you've incorporated what you're infected with. That is a signal to be destroyed. For the antigen presenting cells, the macrophages, the monocytes, those types of things, they will engulf and destroy the bacteria in this case, and they will only put up the MHC class 2 when they have that foreign matter to present, and that's going to activate other immune cells, not to destroy it, but to go out and become activated and find something else. Okay, so this is what we call antigen presentation, the MHC class 1s and the class 2s. Okay. So with those different types of MHC proteins, we have two different types of T cells that can recognize those. So CD8s, CD8 markers are proteins, receptors that are found on the surface of these T cells. A certain group have ones that are called CD8. Another group has CD4. Those are going to be the major differences we're going to talk about. So CD8 cells, these are the ones that communicate and can recognize the MHC type 1 antigen presenting proteins, which makes sense because CD8 cells are also called cytotoxic T cells. They destroy infected cells. So here we have antigen recognition, kind of the step 1. Then we're going to zoom way in. So here we have our infected cell. That's the yellow one down here, right here. And it is presenting its infected antigen in the MHC class 1. Okay, so we just described that on the last slide. The T cell, this cytotoxic T cell, the CD8 cell, has a receptor that has been programmed to recognize that particular antigen. Again, that's the programming that takes place in the thymus. And then here's the CD8 protein, and the CD8 protein is what recognizes the MHC class 1 antigen kind of cup. So it's basically like, here is, I usually do this in class with a handshake, but I can't do that. So here's your antigen, and here's the receptor that's going to recognize it. And then over here, off on the B cell or the T cell, you're going to have the MHC class 1 being recognized by the CD8. Okay, so it's kind of those little lollipop looking things. So once that all happens, you have the recognition of the antigen by the T cell receptor. You have recognition of the MHC class 1 by that CD8 marker. That's going to check all of the boxes. And then you have what's called co-stimulation. It's like a double check on that cytotoxic T cell to say, yes, are you sure that you are infected? Because I'm going to destroy you pretty soon. So once the co-stimulation and all of those other connections, the receptor, the CD8 to the MHC class 1, once all of those are thumbs up, then what that CD8 cell can do is one of these three things, kind of in these boxes. It will destroy the plasma membrane via perforin, very much like the natural killer cell does in immunological surveillance. It will release some chemicals to trigger apoptosis. If you remember from general bio, apoptosis is programmed cell death. These cytokines will trigger this signal transduction inside of this infected cell, and it will destroy itself. Or it could release toxins that will disrupt the cell's metabolism, basically destroying the cell. All right, so that is an infected cell presenting its antigen in MHC class 1, being recognized by the cytotoxic CD8 cells. The receptor recognizes the antigen, the CD8 marker recognizes the MHC class 2, the cell gets activated, and it destroys the infected cell. Okay. Once it is activated by that co-stimulation, we can come back up here to this box, and once it's activated, it will go through many rounds of division and make a whole bunch of these activated cytotoxic T cells. So then they will go around and recognize any cell that's presenting that antigen on MHC class 2 and destroy it. Zoom, 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 like a laser beam, just destroying any cell that presents that MHC class 1. So that's why it's super important to do that co-stimulation to make sure that, yes, this is what I want to fight, because you do not want CD8 cytotoxic T cells just going haywire and destroying cells that aren't really infected. A small portion of those activated cells, or the product of that cell replication, will become our memory cells. So they're not going to be active in the current fight. They're going to hang back as inactive, and then that will be your new population of CD8 cells that will recognize this particular antigen in the future. That's the memory part of our immune response. All right, so that's our activation of CD8 cells by antigen presentation in MHC class 1s. Whew, that's a mouthful. All right. So here is a slide showing a cytotoxic T cell destroying an infected cell. This little YouTube link I have already put on the helpful links in the lymphatic system lecture materials. So you can go and watch that. It's a little micrograph of showing how the cytotoxic T cell comes into direct contact with the infected cell and basically destroys it. So that's a good thing. Okay. This second part of the cell-mediated immunity is the activation of the CD4 cells. So remember the MHC class 2s, the ones that were found on the antigen-presenting cells and other lymphocytes. So they are processing the antigen. They put it up on their MHC class 2. And in this case, we have a receptor, right? So we have the receptor that recognizes the antigen, and here's the CD4 protein that recognizes the MHC class 2. Okay. So this is a T cell that's been programmed to recognize that particular antigen, and it recognizes that it's in the MHC class 2 because of that CD4 marker. So they come into close contact. The antigen presenting cell is like, here, look what I found. Come and help me find some more. The T cell, the CD4 helper T cell comes in, recognizes that antigen, recognizes the MHC class 2. So these are going with your MHC 2s, right? And then once that happens, these aren't destroying cells. These are helper T cells. They're the managers. So they're going to activate and divide. You're going to have a whole bunch of those. A few of those are going to become your memory cells, just like we saw with the CD8s. And then now all of these active helper T cells release a ton of chemicals. These are called cytokines. We're just collectively calling them cytokines. So here you're going to have these cytokines released, and they're going to go and say, hey, natural killer cells, start working harder, and macrophages start working harder, and they can go and activate B cells to make antibodies, and they're going to tell the cytotoxic T cells to become more active. So these cytokines being released from the helper Ts really just ramp up the response of everybody else. So helper Ts, they're called helper for a reason, because they help out, and they're activated by these antigen presenting cells recognized by the CD4 markers on their surface. Right? I think I talked all about all of those things. Let me see. So what do these things do? So they stimulate T cell division, they enhance non-specific defenses, they attract natural killer cells to the infection, and they promote the activation and differentiation of B cells. So that's what all of those cytokines do in an activated T cell. All right. In this little micrograph, I wanted to point out that that orange blob is the helper T, and those little blue circles coming off of it. Anybody? Take a guess. Those are HIV particles. So this is a helper T cell that's been infected with human immunodeficiency virus, HIV. So in this case, the helper T is the infected cell. And so it will, because it's a nucleated cell, present these viral antigens on its MHC class 1. And then you'll have cytotoxic T cells come around and destroy the helper T. And that's one of the signs of converting from an HIV positive person to somebody with the actual disease of AIDS, acquired immunodeficiency syndrome, is because you have not enough helper Ts, because your cytotoxic Ts are destroying and the virus is killing and destroying your helper Ts. So you do not have these managers going around and telling everybody what to do. And so then you have a completely suppressed immune system. And most people who die of AIDS die from general infections that healthy people can typically fight off. Okay, last slide here is just a summary of the differences between your cytotoxic T cells and your helper T cells. So again, in that first column over here, activation by the class 1 MHCs, it is an infected nucleated cell presenting the antigen on MHC class 1. The cytotoxic or the CD8 cells recognize the antigen because that's what they're programmed for. They recognize the MHC class 1, that's the job of the CD8 marker, and then they will destroy that infected cell. That's the activity of the cytotoxic T cells. They will also make their little complement of memory cytotoxic T cells to wait for that appearance in the future. And then also, I didn't mention this when we were going over that CD8 cell activation, but your regulatory T cells, this picture is calling them suppressor, I think the new book calls them regulatory. These are also CD8 marker cells. So they come from this line of cytotoxic T cells, but they kind of get activated a little bit later in the immune response. So when they become active, they kind of send out signals to tell everybody else to chill because the infection's done. All right, and then the second column here with the class 2 MHCs, remember these are presented on antigen presenting cells that may be engulfed or digested a bacteria or a virally infected cell, and they present it up on the MHC class 2 when they're processing that antigenic piece. They are recognized by the CD4 helper T cells, which will then become activated, and the helper Ts will go around and help everybody else increase their immune response. And then they will also do a little collection of memory T cells to await for the next time that pathogen shows up. All right, whew. So that is our cellular mediated immunity with our cytotoxic T cells and our helper T cells, recognizing these antigens presented in these MHC proteins. All right, send questions and on to humoral immunity with antibodies. See ya.